Hello, I'm Theresa Mullin, the Associate Director for Strategic Initiatives at the FDA Center for Drug Evaluation and Research. And on behalf of the FDA and Health Canada, I'd like to welcome you to our virtual regional ICH consultation. Our regional meeting supports the original aims of ICH's reforms back in 2015. And the goals of those reforms included a focus on global pharmaceutical harmonization work in one venue, and that venue would give an opportunity to all key regulatory and industry stakeholders to get more involved in the harmonization work on pharmaceuticals, human pharmaceuticals, and thirdly, to maintain an efficient and well-managed operation so that that guideline work is done in a timely manner and produces excellent quality guidelines. And if you wanted to read more about ICH, all the activities and the articles and procedures that govern its work, there's a link provided for you on this slide. The regional meetings like this one provide us with an opportunity to engage even more with our stakeholders. Since the ICH reforms, we've had growing participation of regulators and industry in that work to harmonize standards for technical and scientific parameters for human drugs. But we've also had an increasing recognition that we need external expertise as well, and engagement and consultation as part of these processes. And we've been looking for expanding opportunities for getting that public input through a variety of means, including expert working groups, in some cases, conduct workshops now to get the other expertise beyond the ICH membership on that guideline work. And we've been publishing reflection papers to get public comment. And these reflection papers are typically focused on proposals for new guideline work in new areas. For example, there was a good clinical practices renovation paper a few years ago, which has now resulted in several streams of work. On this slide, you can see the significant growth that we've experienced in the number and the diversity of ICH participants. The blue font on the left side of the screen shows you the original footprint of the organization prior to 2015, and we had the founding regulatory members and industry members and our standing regulatory members and observers. And since that time, in the green font, you see the tremendous growth that we've had in new regulatory members and industry members, and a large number of observers from legislative and administrative authorities, from regional harmonization initiatives, and from other organizations that are affected by and very interested in and can contribute to the ICH work. Our approach on this is, I'll say, rather like the expression of, the proof of the pudding is in the eating, and the proof or the value of ICH is ultimately in those harmonized guidelines. And so we aim and work hard to make sure that we focus those guidelines on areas that are most relevant and important to regulators and to the industry parties. So we're focusing that work on topics directly relevant to the quality and the efficiency of drug development, regulatory review, manufacturing, post-approval oversight, and other aspects of the life cycle of human drugs. We also focus on scientific rigor, on having data-driven, consensus-based standards, and we do this through work processes that are inclusive and transparent and work very hard to be so. And thirdly, and just as importantly, if not more importantly at the end of the day, as importantly, focusing on implementation. The benefit of these guidelines occurs when they are implemented and when they are used globally. And so here ICH is working on supporting that through training on the guidelines and continued monitoring of the progress of implementation of the guidelines and challenges that any authorities may be facing so that we can understand and try to address that. So for you today, what we'll be covering are a really good cross-section of the work ongoing in ICH. Jill Adleberg is going to give a general overview of ICH, and that'll be followed with Alisa Vespa from Health Canada presenting an overview on guidelines recently reaching some important milestones, and S1 and Q3C will be the focus of that presentation. Khair ElZarrad will be giving a presentation on work ongoing, and in this case will be focused on the ICH E6 GCP principles recently published. Ashley Boam will be presenting on the work on the implementation of guidelines, in this case the ICH Q12. Scott Marshall is then going to present on recent work by the ICH discussion group that's focused on model-informed drug development. And then Robin Bent will provide a presentation on the ICH reflection paper recently issued for comment on patient-focused drug development. And then we'll have time for a session devoted to questions and answers and hearing comments from you participants, all of who have joined us for this meeting. And with that, I want to thank you again for joining us today. We're really very much looking forward to sharing with you and hearing from you on the work that we are doing. Thanks very much.
Thank you for the introduction. For those who may be unfamiliar with the International Council for Harmonization of Technical Requirements of Pharmaceuticals for Human Use, or ICH, I'll provide a brief overview of the organization, its approach, and its process for harmonizing international drug regulatory standards. ICH was established more than 3 decades ago by representatives of the regulatory agencies and industry associations from the US, Europe, and Japan. Health Canada, Swissmedic, and the World Health Organization also provided valuable contributions from the start. ICH fosters development of uniform, scientifically driven standards to improve the efficiency of new drug development and to promote public health by, among other things, preventing duplication of clinical trials in humans and minimizing the use of animal testing without compromising safety and effectiveness. These objectives are achieved by developing harmonized technical guidelines and standards implemented by regulatory authorities around the world. In 2015, ICH made a series of significant organizational changes to extend the reach of harmonization beyond its founding regions. It became a nonprofit legal entity under Swiss law. The reforms were designed to benefit patients around the world by focusing global pharmaceutical regulatory harmonization work in a single forum, by promoting greater involvement from international regulators in global industry sectors, by facilitating greater adoption of new and improved research and development approaches, common standards, and therapeutic advances, and by providing a more stable and efficient operating structure. ICH has grown to include 17 regulatory and industry association members spanning 4 continents and 32 observers, including regulatory authorities, regional harmonization initiatives, international industry pharmaceutical organizations, and others affected by ICH guidelines. Last year, Turkey became ICH's newest regulatory member, and Lebanon its newest regulatory observer. Over its history, ICH working groups have produced and updated approximately 70 harmonized technical guidelines on all aspects of human drug development. ICH has also pioneered development of electronic standards for the transfer of regulatory information, creating an electronic common technical document that brings together information in a harmonized format accepted by regulators in all ICH regions. The electronic common technical document has dramatically reduced the burden on developers seeking marketing approval for a drug product in multiple regions by allowing them to submit data and information organized in the same way to each regulator. ICH has also developed the Medical Dictionary for Regulatory Activities Terminology, or MEDRA, to facilitate sharing of regulatory information internationally, and it has become a global standard in the protection of public health. Measure is a rich and highly specific standardized medical terminology used for registration, documentation, and safety monitoring of medical products, both before and after market entry. Since its launch in 1999, Measure has grown steadily. It's now available in 14 languages and has over 6,800 subscribers in more than 125 countries. This slide provides a sample of harmonized topics considered by ICH working groups. As you can see, the topics range from safety to efficacy to quality to multidisciplinary efforts, including electronic standards. The 70 ICH guidelines previously mentioned cover these and other topics and are based on a wealth of foundational knowledge and experience provided by experts participating on ICH working groups. As you can see from this chart, more than 700 technical experts participated on 34 ICH working groups last year. More than a third of these experts came from recent ICH members and observers, reflecting a significant level of interest and engagement by ICH newest parties. Harmonized guidelines are developed in a 5-step process that begins with adoption of a new topic proposal by the ICH Assembly. Once a concept paper and business plan are agreed to, a working group of experts is formed to develop the guideline, and work begins in earnest. The expert working group develops a consensus draft of the guideline. When the draft is completed, the topic leaders for each party represented on the working group signs off on the guideline at step 1. Step 2 is a 2-step process. First, the Assembly endorses the draft guideline. Second, regulators separately do so. The guideline then moves to a public consultation phase, in which it's submitted for public comment in a transparent process designed to solicit thoughts from a wide array of stakeholders. Members solicit public comments from the regions. The draft guideline is also posted on ICH's website for others to comment. The expert working group collects all of the comments, reviews and evaluates them, and produces a final guideline. At step 3, the working group leaders sign off on the final guideline. At step 4, the ICH Assembly and its regulatory members approve the final guideline. And finally, at step 5, ICH regulatory members and observers implement the final ICH guideline within their respective country or region, achieving the goals of harmonization. To enhance adoption of harmonized standards, ICH develops training materials based on its guidelines. These include a training library available on ICH's website, providing funding for training offered by ICH regulatory members and observers, recognized training programs hosted by a variety of organizations, and other online training materials, including some translations. ICH is governed by the Assembly, including all ICH members and observers. The Assembly approves new topics for harmonization, adoption, amendment, or withdrawal of guidelines, admission of new members and observers, and election of Assembly and Management Committee leaders. The Management Committee is a smaller body comprised of permanent and standing members and elected members that oversees operations including administrative and financial matters and oversight of working groups. It recommends to the Assembly selection of new topics for harmonization and adoption, withdrawal, or revision of guidelines. It also includes a Finance Committee that develops ICH budgets and allocates funding. This slide maps the ICH governance structure. At the center is the Assembly. The ICH and Measure Management Committees branch to each side. The Secretariat provides day-to-day management of ICH, including preparing and documenting Assembly and Management Committee meetings. ICH coordinators are selected by each member and serve as a central contact for their organizations, with the Secretariat and facilitate conversation between the ICH Management Committee and the working groups. To be approved for ICH membership, a regulator must be a recognized authority responsible for regulation of human drugs. They must have demonstrated engagement in ICH through regular attendance in ICH meetings and participation in working groups, and they must have implemented at least 3 foundational guidelines, including Q1, Q7, and E6. To be approved for ICH membership, industry organizations are required to represent members from several countries in at least 3 continents regulated by ICH guidelines, and like their regulatory counterparts, have demonstrated engagement in ICH through regular attendance in ICH meetings and participation in working groups. To wrap up, ICH is a unique organization that draws on the expertise of regulators in industry to achieve international harmonization. It uses a transparent, science and consensus-based process to develop guidelines that includes opportunities for public comment. It includes a commitment by regulators to implement harmonized guidelines, and its expanded global participation and engagement through recent reforms. I hope this introduction has been helpful. Thank you very much for your time, attention, and interest in our efforts.
Good afternoon, and thank you for the introduction. This is the order and outline of the presentation. I will first provide an update on the ICH Q3C guideline for residual solvents, where monographs and permitted daily exposure values have been developed for 3 new solvents. Following this, I will present an overview of the draft addendum to the ICH S1B guideline on carcinogenicity testing, which expands the testing scheme for assessing human carcinogenic risk of small molecule pharmaceuticals beyond what is currently recommended in the guideline. Beginning with the ICH Q3C guideline for residual solvents, monographs and permitted daily exposure values have been developed for 2-methyl tetrahydrofuran, cyclopentyl methyl ether, and tertiary butyl alcohol. The purpose of the ICH Q3C guideline is to recommend acceptable levels of residual solvents in pharmaceuticals to ensure patient safety. Within the context of ICH Q3C, the acceptable levels are termed permitted daily exposures, or PDEs. The ICH Q3C core guideline was adopted by ICH in June 1997. Then, in 1999, a maintenance expert working group was formed to allow for revising existing PDEs as new toxicity data becomes available, and also to develop monographs and derive PDEs for new solvents when adequate toxicity data is available. There is a long history of using the maintenance working group for these purposes, and in the past 20 years, the Q3C guideline has undergone several revisions. In May 2017, consensus was reached to include 3 new solvents in the Q3C guideline, and they are 2-methyl tetrahydrofuran, cyclopentyl methyl ether, and tertiary butyl alcohol. The maintenance working group developed a monograph and a PDE for each solvent, and once consensus was reached by the working group on the Step 1 draft document, it was submitted to the ICH Assembly, where it was endorsed in March 2020. Soon after, the monographs entered Step 3 of the ICH process, which consists of regulatory consultation across the ICH regions, expert working group discussion of comments received, and document revision as necessary. Step 3 concluded in April 2021 when consensus was reached on the revised guideline. As part of Step 4 of the ICH process, the guideline was submitted to the ICH Assembly and adopted as a final harmonized guideline in April 2021. This version constitutes the 8th revision of the guideline and is coded as ICH Q3C R8. It is now posted on the ICH website, and the link is available at the end of this slide. I will now provide a summary of the toxicity data for each of the three solvents and also outline how the PDE for each solvent was derived. In the case of 2-methyl tetrahydrofuran, or 2-MTHF, this compound displayed no evidence of genotoxic potential. Carcinogenicity data was not available, and there were no reliable reproductive toxicity studies for PDE calculation. With respect to repeat dose toxicity, 2 3-month oral rat studies were available, and one of the studies was considered appropriate for calculating a PDE. In that study, groups of male and female rats were orally dosed with 2-MTHF at 80, 250, 500, and 1,000 milligrams per kilogram once daily for 3 months. Based on slight effects on kidney weights and prothrombin time at 500 milligrams per kilogram, the no observed effect level, or NOAL, was considered to be 250 milligrams per kilogram per day, and this value was used to calculate the PDE. The calculation consists of multiplying the NOAL by a weight adjustment of 50 kilograms and then dividing by the appropriate safety or modifying factors F1 through F5 as recommended in the Q3C guideline. Details on the modifying factors applied in the case of 2,8-MTHF can be found in the guideline. Carrying through with the calculation, a PDE of 50 milligrams per day is derived. As a result of the Step 3 regulatory consultation, the monograph for 2-MTHF was revised to include results of an OECD 414 and GLP-compliant rat developmental toxicity study. Inclusion of this study in the monograph did not alter the recommended PDE of 50 milligrams per day. Therefore, this is the final PDE for 2-MTHF, and per the classification system outlined in the ICH Q3C guideline, this solvent is placed into class 3 as it exhibits low toxic potential. Moving on now to cyclopentyl methyl ether, or CPME, this compound displayed no evidence of genotoxic potential. Carcinogenicity data were not available, and there were no reliable reproductive toxicity studies for PDE calculation. With respect to repeat dose toxicity, 2 oral rat studies were available, one 28-day and the other 90-day, and a 90-day inhalation study, also performed in rats, was available. Following an assessment of study design and level of information provided in each study report, it was determined that the 28-day oral rat study was considered the most appropriate for calculating a PDE. In this study, male and female rats were orally dosed with CPME at 15, 150, or 700 milligrams per kilogram per day for 28 days. Findings included unscheduled deaths in males at the high dose of 700 milligrams per kilogram per day, which was attributed to poor clinical condition. Other adverse effects observed in the high dose group included decreased motor activity, tremors, and convulsions in males, and decreased body weight gain in females. There were no toxicologically significant findings at 150 milligrams per kilogram per day, and this dose level was considered to be the NOAL. To calculate a PDE, the NOAL of 150 milligrams per kilogram per day was multiplied by a weight adjustment of 50 kilograms and divided by the appropriate modifying factors F1 through F5. The calculation is performed and a PDE of 15 milligrams per day is derived. As a result of the Step 3 regulatory consultation, minor editorial revisions were made to the monograph, and there was no change to the recommended PDE of 15 milligrams per day. Therefore, this is the final PDE for CPME, and this solvent is classified as a Class 2 solvent to be limited. The last solvent is tertiary butyl alcohol, or TBA, and substantially more data is available for this solvent when compared with the other two solvents. Moving through the data, there was no evidence of genotoxic potential, and there was some evidence of TBA-induced reproductive and developmental effects at the maternal dose of 1,000 milligrams per kilogram per day, and includes increased pup mortality and increased number of stillborn pups. The no observed adverse effect level for reproductive and developmental effects was 400 milligrams per kilogram per day. With respect to repeat dose toxicity, 2 13-week drinking water studies are available, one in rats and the other in mice. In the rat study, mortality was observed at the highest dose level tested, and adverse effects were noted in the kidney, consisting of nephropathy, as well as the urinary bladder, consisting of inflammation in both males and females. A no observed effect level was not identified in this study, and the lowest observed effect level, or LOEL, was 176 milligrams per kilogram per day, based on nephropathy in males. In the mouse study, mortality was observed at the highest dose level tested, and adverse effects were noted in the urinary bladder of both sexes and consisted of transitional epithelial hyperplasia. The NOEL was considered to be 1,786 milligrams per kilogram per day based on this microscopic finding. Finally, the carcinogenic potential of TBA was investigated in rat and mouse drinking water studies conducted by the U.S. National Toxicology Program, or NTP. The primary targets of both toxicity and carcinogenicity were the kidney in rats and the thyroid and urinary bladder in mice, and the NTP concluded that there was some evidence of carcinogenic activity in male rats and female mice. Following an assessment of study design and quality of the study report, the working group concluded that the 2-year carcinogenicity studies were the most appropriate for calculating a PDE. For this reason, a point of departure was determined for each study and 2 PDEs were derived. Since the PDE calculated from the mouse study was greater than the one calculated from the rat study, the PDE outlined in the Q3C document was derived from the rat study as it represents the more conservative value. I will now describe the 2-year rat study in a little bit more detail. In this study, male and female rats were orally dosed with TBA by drinking water at 85, 195, and 420 milligrams per kilogram per day in males, and 175, 330, and 650 milligrams per kilogram per day in females for 2 years. In males, an increased incidence of renal tubule adenomas and carcinomas were observed, but the increase was not dose-dependent, and the tumors were not considered human relevant, as the evidence suggests that they likely formed through an alpha-2U mediated mode of action, which is male rat specific. In females, there were no remarkable neoplastic findings. However, a dose-dependent increase in severity of nephropathy was observed at all dose levels. As a result of this finding, the lowest observed effect level for the 2-year rat study was considered to be 175 milligrams per kilogram per day. To calculate a PDE, the LOEL was multiplied by a weight adjustment of 50 kilograms and divided by the appropriate modifying factors F1 through F5. The calculation is performed and a PDE of 35 milligrams per day is derived. As a result of the Step 3 regulatory consultation, minor editorial revisions were made to the monograph, and there was no change to the recommended PDE of 35 milligrams per day. Therefore, this is the final PDE for TBA, and this solvent is classified as a Class 2 solvent to be limited. As previously mentioned, these 3 new monographs have been finalized and are now available on the ICH website. And that brings me to the end of the Q3C update. The second part of this presentation will provide an overview of the addendum to the S1B guideline on testing for carcinogenicity of pharmaceuticals. The purpose of ICH S1B is to provide guidance on approaches for evaluating the carcinogenic potential of pharmaceuticals. This guideline was adopted by ICH in July 1997. Currently, carcinogenic potential can be evaluated using option 1 by conducting a 2-year carcinogenicity study in one rodent species and a short or medium-term in vivo study in a second rodent species, or using option 2 by conducting a 2-year carcinogenicity study in both the rat and the mouse. When option 1 is utilized, the approaches cited on this slide have become the most widely used. That is, a 2-year rat study is conducted alongside a short-term study using a transgenic mouse model of carcinogenesis, such as the RAS-H2 model. Since the time the S1B guideline was published in 1997, several advancements have been made, including better understanding of the mechanisms underlying tumor formation in rodents, which can inform whether these findings are relevant to human carcinogenic risk. In addition, a number of retrospective analyses have been conducted which suggested that the outcome of the 2-year rat study did not further inform human carcinogenic risk of the pharmaceutical based on a comprehensive assessment of available biological, pharmacological, and toxicological data. While the retrospective assessments were considered valuable and informative, it was unclear whether an evaluation of the available data would be sufficient to prospectively conclude that a 2-year rat carcinogenicity study does not further inform human carcinogenic risk. For this reason, the expert working group proposed to conduct a prospective evaluation study to better define the factors that should be included in this comprehensive assessment, also known as a weight of evidence assessment, and also to determine the set of criteria that could support a conclusion that a 2-year rat study would not add value to the assessment of human carcinogenic risk with a reasonable degree of certainty. The concept paper and business plan describing the expert working group's proposal was developed and finalized in November 2012. And the prospective evaluation study was launched in the year 2013. There is insufficient time in this presentation to go over the design and results of this study, and so I refer you to the ICH website where the regulatory notice document, or R&D, is posted that provides details on the objectives and design of the prospective evaluation study. Several status reports are also posted that provide a high-level summary of data obtained at specific points in time. Between the years 2013 and 2021, the study was carried out. Data was accrued, analyzed, and discussed, leading to the development of a Step 1 draft addendum, and consensus was reached by the expert working group in March 2021. The draft addendum was recently endorsed by the ICH Assembly in April 2021, and Step 3 of the ICH process, which consists of regulatory consultation across the ICH regions, will begin in the near future. I will now provide an overview of the content of the addendum. The purpose of the S1B addendum is to expand the testing scheme for assessing human carcinogenic risk of small molecule pharmaceuticals by including a weight of evidence approach to determine if the 2-year rat study adds value to the assessment. It is important to note that the addendum does not replace the existing S1B guideline, and the Option 1 and Option 2 testing schemes discussed a couple of slides ago remain an important part of the carcinogenicity testing paradigm. The addendum also adds a plasma exposure ratio endpoint for high-dose selection in the RAS-H2 transgenic mouse model. this pharmacokinetic endpoint can be considered in addition to the other high-dose endpoints described in the S1C guideline. Beginning with the first objective of the addendum, it is outlined in the document that the conduct of a weight of evidence assessment may lead to one of three possible conclusions. First, based on the available data, the pharmaceutical may be considered likely to be carcinogenic in humans. In this case, the outcome of a 2-year rat study would not add value to the assessment of human carcinogenic risk. Or it may be concluded that a pharmaceutical is not likely to be carcinogenic in humans, and the conduct of a 2-year rat study would similarly not add value to the assessment of human carcinogenic risk. The third possible conclusion is that the carcinogenic potential of a pharmaceutical in humans is uncertain. In this case, a 2-year rat study will likely add value to the assessment of human carcinogenic risk. So what factors should be evaluated in the weight of evidence assessment to determine the direction of the carcinogenicity testing plan? 6 factors are outlined in the draft S1B addendum. Data supporting the assessment of each factor can be obtained from public sources and also from conventional drug development studies, and should be relevant to the evaluation of carcinogenic potential. The first factor is an understanding of potential carcinogenic risk based on drug target biology and the primary pharmacologic mechanism of the parent compound, as well as any major human metabolites. Assessment of this factor will likely consider information from various sources, and examples are outlined in the addendum, which includes carcinogenicity data from other compounds in the drug class. The second factor to evaluate is off-target potential, which can include the results of secondary pharmacology screens for the parent compound and major metabolites. Of note, assessment of off-target effects relevant to potential carcinogenic risk may be considered particularly informative and can include, for example, a nuclear receptor screen. The third factor consists of an evaluation of histopathology data from repeat dose toxicity studies. The outcome of the prospective evaluation study indicates that histopathology data from the long-term rat toxicity study is the most informative for assessing the value of the 2-year rat study. For identified and relevant effects, such as cellular hyperplasia, it is important to demonstrate understanding of the pathogenesis and also determine whether the effect is considered human relevant. Included in this assessment is calculation of exposure margins for both the parent compound and any major metabolites. The fourth factor consists of evidence of hormonal perturbation. Sources of data for this factor are various and can include histopathology findings in endocrine and reproductive tissues, as well as the results of reproductive toxicology studies. As with the third factor, findings suggestive of potential carcinogenic risk should be investigated to determine human relevance and/or understanding of the mode of action. The fifth factor consists of genetic toxicology data derived from studies outlined in the ICH S2 guideline on genotoxicity testing. And the final factor consists of evidence of immune modulation, which can include results from studies outlined in the ICH S8 guideline on immunotoxicity. While comprehensive assessment of these 6 factors may be sufficient to conclude that a 2-year rat study is or is not likely to add value to the assessment of human carcinogenic risk, there may be cases where additional investigations may be needed to better inform human relevance of a potential risk. This may include the conduct of additional investigative studies or analysis of specimens collected from prior studies. In other cases, availability of clinical data may be useful to determine if the presumed mode of action in rodents is relevant in humans at therapeutic exposures. The draft addendum provides some examples of data that may facilitate the de-risking of inconclusive findings or those which suggest cause for concern. As alluded to in the previous slides, it is the integration of the available data supporting each weight of evidence factor that will determine whether or not a 2-year rat carcinogenicity study will add value to the assessment of human carcinogenic risk. How this integrated assessment may be accomplished is outlined in the form of case studies drawn from the prospective evaluation study and are presented in Appendix 1. Also highlighted in Appendix 1 are the characteristics of a compound that are more likely to support a conclusion that a 2-year rat study will not add value. Experience from the prospective evaluation study indicates that carcinogenicity data from other compounds in the drug class contribute significantly to the weight of evidence, as it constitutes a valuable data set informing whether modulation of the drug target is or is not associated with a potential carcinogenic risk. Therefore, while novel drug targets or first-in-class compounds are eligible for a weight of evidence approach, a higher evidentiary standard is expected to demonstrate no cause for concern in regards to target biology and primary pharmacologic mechanism. Of note, Appendix 1 includes 2 case studies for first-in-class compounds. Importantly, a mouse carcinogenicity study in the form of either a 2-year study or a short-term transgenic model assay remains a recommended component of the carcinogenicity testing plan, even if the outcome of the weight of evidence assessment indicates that a 2-year rat study does not add value. That said, an exception to this recommendation can be made for drug submissions in the European Union, specifically when the weight of evidence assessment indicates that a 2-year rat study does not add value. A mouse carcinogenicity study is also not recommended. So that is the end of the presentation on the weight of evidence approach being proposed in the draft S1B addendum. And I will now switch gears and briefly discuss the addition of a plasma exposure ratio endpoint for high dose selection in the RAS-H2 transgenic mouse model. The ICH S1C guideline on dose selection for carcinogenicity studies indicates that the high dose can be selected by a variety of approaches, including application of a limit dose, consideration of pharmacodynamic endpoints, toxicity endpoints, as well as pharmacokinetic endpoints, in which the S1C guideline indicates that a 25-fold plasma AUC exposure ratio can be used in 2-year rodent studies. As the analysis that supported this recommendation did not include the outcome of RAS-H2 transgenic mouse studies, use of the 25-to-1 plasma AUC exposure ratio endpoint was not considered acceptable for this transgenic mouse model. For this reason, a retrospective assessment of RAS-H2 transgenic mouse studies was performed by Hisata and colleagues. The data indicate that while a low number of pharmaceuticals were positive for carcinogenicity at doses that exceed a 25-fold plasma AUC exposure margin, positive outcomes were not observed at a 50-fold plasma AUC exposure margin. Therefore, it was concluded that there is no value in exceeding a 50-fold ratio for carcinogenicity studies conducted in RAS-H2 transgenic mice. Summary data supporting the proposal for inclusion of a 50-fold plasma AUC exposure margin as a high-dose pharmacokinetic endpoint in the RAS-H2 mouse is outlined in the draft addendum, and it is anticipated to be published in the near future. Of note, this high-dose endpoint does not apply to other transgenic models of carcinogenesis. As part of the next steps, public consultation on the draft S1B addendum will soon be initiated in the ICH regions. The regulatory agency in each ICH region is responsible for opening a consultation period and soliciting comments on the draft document. Within the context of the ICH regions represented at this meeting, comments can be submitted to Health Canada at the email address outlined on this slide, or to the US FDA at www.regulations.gov once the addendum has been published in the Federal Register. When the public consultation ends in all the ICH regions, comments will be collated and discussed by the working group, and revisions will be made to the addendum where warranted. Finalization of the addendum as a Step 4 document is planned for the summer of 2022. This is my final slide, and I would like to acknowledge members of the ICH Q3C and ICH S1 expert working groups, as all of the information presented here are work products of these groups. Thank you for your attention, and I will now answer any questions.
Hello, everybody, and thank you so much for this opportunity to present to you. My name is Khair, ElZarrad. I'm the Deputy Director for the Office of Medical Policy at the Center for Drugs at the US FDA. I'm also the rapporteur for the ICH E6R3 Expert Working Group, or EWG. I'm delighted today to present to you some updates on the development of R3, the guideline for good clinical practice. For today, I'm hoping to provide you with a little bit of perspective on the rapidly evolving evidence generation ecosystem as the backdrop for a lot of the work we're doing around GCB and clinical trial conduct in R3. And then following that, I'll provide a description of the ICH E6R3 Expert Working Group approach, and that will include three levels: the development strategy, the analysis of the public input that we've done over the last few months, and then the stakeholder engagement, which I believe to be a very important and unique aspect for the development of E6R3. I will follow with a little bit of an overview of the published draft E6R3 Introduction and Principles. We published the draft Introduction and Principle around 2 weeks ago, and we're very excited about them, and I'm going to give you a quick overview of what we published. I will end with a summary, but also an invitation to the EWG web conference on May 18th and 19th next week. We planned this conference that's focused on E6R3 to further present the principles and our work, but also to hear from stakeholders on their vision and aspiration for GCB and the clinical trial enterprise in general. So we will be delighted to have you there. Just as a backdrop for the EWG work, we were very aware of the advancing evidence generation paradigm. The multiple streams of data, as shown in that figure, the big data figure, and also the real-world data that's available increasingly to us to consider its potential. This was highlighted in the real-world evidence program from the FDA, the one you see in front of you, that explores the potential utility of real-world data to help us in decision making. Match that with an increasingly digital world. I know many of us are familiar with sensors, with trackers, but this goes far beyond that. You see multiple innovations around the use of AI, artificial intelligence, and the use of remote data capture, and multiple other tools, passive sensing, that's really going to help us, I believe, provide us with a tremendous opportunity here to make clinical trials more efficient. It's important also to mention the innovations in clinical trial designs, and to highlight just a couple. Decentralized clinical trials provide the opportunity to reach patients in their own communities, in their own homes, potentially. And you can imagine how that really can be helpful whenever that's appropriate to utilize. Also, the pandemic over the last year or so that really catalyzed our thinking around the efficiency in clinical trials and how, for example, we can incorporate clinical trials in healthcare settings. So all of this is really the backdrop, the thinking of the EWG, and how can we address the opportunities here, and facilitate it will also ensure the reliability of the results from clinical trials and the protection of the participants. Many of us are aware of the history of E6, but just to highlight a couple of points, the original version was finalized in 1996. It describes the responsibilities and expectations of stakeholders in the conduct of clinical trials. It covers aspects of monitoring, reporting, and archiving clinical trials. Now there is R1, revision 1, that has minor updates to it, but the current version is R2 that was finalized in 2016. It included an addenda with multiple updates, including an addendum to encourage implementation of more efficient GCP approaches and an updated standards for electronic records. This is the background that the EWG have to start working from. A quick update on or review of the approach that the EWG is taking to develop E6R3. We're thinking of the principles as overarching and as remaining to be relevant as the technology evolve, as design advance. And that box in front of you highlight those principles. We have also Annex 1 that's going to complement the principles, and that's Annex 1 addresses the GCP for interventional clinical trials, which will reflect what's really currently in the concepts in the R2 version. And that will be also with updates, refinements, and changes informed by our analysis and engagements and thinking as EWG. Now, we understand that Annex 1 will also be relevant and applicable to other clinical trial designs that we consider non-traditional clinical trial designs, for example, decentralized clinical trials. However, there may be certain areas that require additional clarifications, and this is why Annex 1 is really important. You see the overlap, highlighting again what I said, that Annex 1 will still be applicable to those non-traditional interventional clinical trials and address many needs around that. However, Annex 2 will provide any added details that are needed. So the approach to E6R3 development is simultaneously working on the principles and the annex, as you see here in the big two arrows, the principles and the annex. And we're currently in this stage of development. And we're hoping that after the principles and the Annex 1, which constitute the replacement of the current R2, reach step 1 and for public consultations, the feedback from the public consultation will also help inform us in the refinement of Annex 2. It's important to know that the current work on principles in Annex 1 is continually informing us on what should be the content of Annex 2. And that is important because, as you see, we have to develop a new concept paper for Annex 2, make sure the ICH Management Committee is also aligned and approving of our thinking for Annex 2. It's important to highlight also that we published, as I mentioned a couple of weeks ago, the draft work in progress introduction and principles, and I believe that's a unique step, but it's really an important step to enhance transparency in our work in general. So the work on E6R3 development is informed by the results of an extensive analysis of stakeholder input and by consistent engagement with stakeholders. The working group conducted an extensive analysis that actually had two prongs to it, two approaches. The first is an analysis of stakeholder comments on E6R2. Practically everything available to us that come from public input, from literature, as I'm going to mention in a few minutes, we looked at that. And also it's an analysis of select ICH guidelines to help align between relevant guidelines wherever that is appropriate. The goals of this analysis is to identify opportunities for improvements in E6R3 and to provide the EWG with potential ideas, potential options on how and where to apply those modifications, practically to inform our work in general. So this is just to highlight samples of the resources used to inform this analysis. We looked at academic responses, open letters, published articles. We also looked at an extensive survey that's done by the Clinical Trials Transformation Initiative, or CTTI, to inform the innovation of ICH E6. And they did actually performed an extensive survey and in-depth interviews and provided multiple comments. And that was one of the resources we've used for our analysis. We also looked at public engagement materials. We conducted multiple regional meetings, the American engagement meetings and Europe engagement meetings. This is all around E6, to enrich the discussion of the EWG. And of course, the Japan engagement meeting as well. And resources from those meetings are available, and we looked at those. For the guideline, ICH guideline analysis, we looked at all efficacy guidelines and also at M11 and any available peer-reviewed publications that also lend themselves to looking at ICH guidelines and analyzing them and providing us with some feedback. So as you can see, it's as comprehensive as possible, the analysis that was conducted by working group members. This is just examples of the areas that were identified for potential updates. Data management is a key issue, considering the digitization of the data ecosystem that we've discussed, and how we're going to look at that from a GCB perspective. The responsibilities is another important area, as we see variable roles, variable expertise potentially being involved in the trial as trials become more integrated in healthcare settings, for example. The clarity is an important issue here, so this is an important area. Monitoring, considering highlighting further the importance of risk-based approaches to encourage risk-based approaches and the variety of monitoring approaches that could address the need. Moving to engagement, to highlight that engagement is essential to inform the EWG work, and acknowledging the wide impact of E6 and the many stakeholders who are affected by this guideline. The ICH Management Committee approved an engagement plan, and I put the link for that below for the EWG, and we're very grateful for that. The engagement plan includes public meetings, such as conducting web conferences, and as I mentioned, we conducted the regional web conferences to support and enrich the discussion of the EWG, and also publishing updates, such as the one we've seen a couple of weeks ago when we published the draft in progress introduction and principles. The web conference, actually, the one I mentioned to you all and invited you to on May 18th and 19th is part of this effort. However, more consistent engagement track we have also as part of this engagement plan is direct engagement with academic experts, selected academic experts during the EWG meetings as the work on the guideline proceeds. We found this engagement so far to be extremely fruitful and useful to us, and we were hoping to continue it as we move forward in the development of guidelines. This is just an acknowledgement of the experts from different regions who are involved in the work, and then we're very grateful to their contributions. And as you can see, they're representative of different regions and providing different and rich expertise to us. So before we jump into the introduction and the principles, I just want to kind of remind you again what we were talking about here. I've showed you this figure before, and what we're talking about is the box here, the draft introduction and principles published in April 2021. We hope those principles will be comprehensive and remain relevant as technology evolves and clinical trial design advances, leveraging and facilitating an increasingly digital ecosystem. Risk-based approach and proportionality will be stressed, and then thoughtful process throughout the clinical trial conception, design, and conduct and analysis will be highlighted. The introduction is an integral part of the E6R3 document, and we were trying to highlight that. We looked at it as more of a manual on how to read and how to utilize the content of the guideline. I'm going to show you a little bit of the content of the introduction in a bullet format, just to give you an idea of what we're trying to highlight. Of course, we highlight that clinical trials are a fundamental part of the clinical research that support the development of new medicines or uses of existing medicine. We stress the importance of clinical trials in multiple locations to highlight their importance. And the principles for GCB are designed to be flexible and applicable to a broad range of clinical trials. The principles of R3 in general are being developed to encourage thoughtful considerations and planning to address specific and potentially unique aspects of an individual clinical trials. Practically, you have to consider all the characteristics of your clinical trial when you're thinking about the conduct and GCB in general. The principles are intended to support improved and more efficient approaches to trial design and conduct. For example, innovative digital health technologies may expand the possible approaches to trial conduct. Such technologies can be incorporated in existing healthcare infrastructure and enable the use of a variety of relevant data sources in clinical trials. And you can see how that ecosystem that I discussed has been reflected in our work. In the introduction as well, we mentioned that the use of technology in the conduct of clinical trials should be adapted to fit the participation characteristics and the trial design. I think this is important, and we were trying to say here is that just by having a flashy technology is not good enough practically. You really have to have the technology answering a need in the clinical trial and fitting within the designs and the characteristic of that trial. The use of innovative technologies may help enable those designing and conducting a trial to include relevant patient population. And the process of building quality into the design of the trial may be supported by participation of those directly involved. And these may include a broad range of stakeholders, including patients and treating physicians. The guideline is intended to be media neutral to enable the use of different technologies for the purposes of documentation. Clinical trials should be designed to protect the rights and safety and well-being of participants and assure the reliability of the results. You see that as the key paradigm, actually, of what we are highlighting as the main purpose of having a GCV is those two factors, the protection of participants and the reliability of results. Clinical trial designs and processes should be proportionate to the risks inherent in the trial and the importance of the trial being collected. And trial designs and processes should be evaluated to minimize unnecessary complexity and burden. So jumping into the principles are overarching, as I mentioned, and provide a flexible framework for clinical trial conduct. They are structured to provide guidance throughout the life cycle of the clinical trial. And these principles are applicable to trials involving human participants, i.e. healthy volunteers or patients. The principles are interdependent and should be considered in their totality to assure ethical trial conduct and reliable results. And the EWG actually discussed the ordering of the principles, or even categorizations and grouping of them. And we increasingly realize that there is an interdependency here, and the principles are really important in their totality, and we try to reflect that here. So a couple of things to highlight here too, that although R3 principles are built on the current principles found in R2, they provide additional elaboration and information to support the intended purpose of R3 principles being comprehensive. And the added clarity also supports the goal that these principles are intended to remain relevant as technology, design, and methods evolve. Although there is an alignment, as I said, between R2 and R3 principles, you will notice differences between them. You know, those range from changes such as the using of the term participants instead of patients, to specific changes intended to provide the needed flexibilities to encourage the use of innovative technologies, innovative designs, while at the same time ensuring the safety of trial participants. Each principles, by the way, have multiple sub-bullets and multiple details underneath it. I'm not going to show you that. That's available publicly, and there's already too many words on my slides. So what I'm going to do, I'm going to show you the principle and provide you with some key points that the EWG discussed about them. The principle number one is the highlighting that the ethical principles that we are aligning with the ethical principles that have their origin in the Declaration of Helsinki. And this is the same principle that exists in R2, and it just again highlight that the coming together, the alignment around the ethical principles in the Declaration of Helsinki. This principle has no additional explanation. I think it's self-explanatory, so you will see it stand alone as our first principle. The second principle highlight that clinical trials should be designed and conducted in ways that ensure the right safety and well-being of participants. The EWG wants to make it unambiguous that the right safety and well-being of the participants are the most important consideration here. And the safety of the participant is not a static thing, and it should be reviewed periodically. The EWG also highlighted that the trial should be initiated and continued only if the anticipated benefits justify the risks. And the EWG wanted to include language to discourage unnecessary exclusion from clinical trials. And also, the EWG discussed that although a qualified physician or a dentist are responsible for the medical care given to or the medical decision made on behalf of the participants, other qualified professionals can also provide care and interact with patients as part of the clinical trial. And as clinical trials are incorporated in healthcare infrastructure and designs, such as decentralized clinical trials are considered, a variety of qualified professionals may be involved. And you can see how across the board we're trying to make the logic around that. Principle 3 is on informed consent, and it highlights, of course, that the freely given informed consent should be obtained, and that informed consent process should take into consideration the relevant aspects of the trial, such as the participants, the risk of the medical intervention, and the setting and the context of the trial, in which the trial will be conducted. And the example we're thinking about is trial in emergency situation, for example. Very relevant. And then the potential for the use of technology to inform participants and obtain informed consent should be also explored. Again, we've seen that how in certain circumstances technology can really facilitate the informed consent process. Principle 4, clinical trials should be subject to objective review by an IRB or an IEC. Of course, IRB review is essential as the trial should be conducted in compliance with the IRB reviewed protocol. And we highlighted also that periodic review of the trial by the IRB or the IEC should also be conducted as appropriate here. Principle 5 highlight that clinical trials should be scientifically sound for their intended purpose and based on robust and current scientific knowledge and approaches. Clearly, we owe it to clinical trial participants and to all of our stakeholders to make sure clinical trials are based on robust science. And as science evolves, periodic look to ensure that any adjustment are applied is also might be necessary. Principle 6 states that clinical trials should be designed and conducted by qualified individuals. Those involved in the design and conduct trial, of course, should be qualified to perform their respective tasks. And in this principle, the EWG highlighted that the trial design, as trial design evolve and technology is being utilized, there are a variety of experts, as I mentioned before, that may be parts of the trial. The EWG discussed also within this principle that training shouldn't be a blanket requirement, but it should be responsive to the expected role within the trial. Principle 7 highlight that quality should be built into the scientific and operational design and conduct of the clinical trial. This highlights that quality is essential across the life cycle of the trial. Efforts and resources should be focused on factors critical to the quality of the trial. And those factors are characteristics that are fundamental to the protection of the participants and the reliability and interpretability of the trial results. Quality of clinical trial is considered in this document as fit for purpose, and the quality and amount of information generated during the clinical trial should be sufficient to support good decision making. Building quality into a clinical trial also means that approaches are developed and implemented to detect and manage any issues potentially. In addition, those approaches and to prevent the reoccurrence of such issues are important to be developed and implemented. This is all highlighted in the details under this principle. Principle number 8 mentioned that clinical trial processes, measures, and approaches should be proportionate to the risks to participants and to the reliability of trial results. Proportionality is actually a key concept that the EWG wanted to strongly encourage across clinical trial processes. It highlighted that clinical trials should be efficient and the focus of the efforts and resources should be proportionate to the risks to the participants and the reliability of results. One key item that highlighted by the EWG also is that risks beyond those standard of care should be the focus of consideration. However, the risks relating to the interventional product which have a marketing authorization used in a clinical context may differ from the usual care of patients and should be taken into considerations. So even if you have an intervention that's already have a marketing authorization that's used within the context of clinical trials, the risks within that context also should be considered. Principle number 9 highlights that clinical trials should be described in a clear and concise and operationally feasible protocol. The clarity and avoiding, of course, unnecessary complexities will not only make a trial more feasible, but it will also conserve important efforts and allow for better focus on what really matters most. This is specifically important in the protocol considering the centrality to the conduct of its centrality to the conduct of the trial. Being clear and concise goes a long way, of course, in making clinical trials less complex and more patient-centric. Principle number 10 highlights that clinical trials should generate reliable results. As you're all aware, clinical trials are substantial undertaking that require many resources, many efforts, and it requires that precious time and efforts of the participants be also invested. Therefore, it's essential that trials are designed and conducted to generate reliable results. That's ultimately really the goal. And all the systems of the processes that involved in the trial should help ensure the quality of the information generated from the clinical trial. And the way those processes and systems are implemented should be proportionate to the risks to participants and reliability of trial results. Again, hitting at the concept of proportionality across the board. Validation of system, data protections, information technology system, and user management are all important elements that should be addressed to ensure the trial generates reliable data. And as we witness the expanding digitization of the data system, appropriate management of data integrity, traceability, and protection of personal information are all important consideration here. And this will help us ensure the accurate reporting, interpretation, and verification of the clinical trial information. The AWG finally here highlighted too that the importance of transparency, which include a registration on a publicly accessible database. Principle 11 highlights the rules and tasks and responsibility in clinical trials should be clear and documented appropriately. And as I mentioned before, as more trials utilize healthcare infrastructure or incorporate innovative technology, and such as the use of decentralized designs, this will enable a wider reach to participants, but it will likely also include or require the involvement of multiple experts and professionals. So clarity of tasks, delegations, and who's doing what practically is really important. Our last principle highlight that the investigational products used in a clinical trial should be manufactured in accordance with applicable good manufacturing practices. Standards and be stored, shipped, and handled in accordance with the product specifications and the trial protocol. Again, as we discussed before, as we see clinical trials being designed increasingly to reach patients, with decentralized module in them, it's important to not only think of the investigational product manufacturing, but also think of the storage, the shipping, and handling, practically the logistics around the use of these investigational products. And measures should be in place to ensure that the investigational products provided to trial participants retain its intended quality here. So all of this also should be considered from the point of view of being proportional to the risks involved. With that, I would like to end with a few bullets here highlighting that well-designed and conducted clinical trial are essential. And this is the goal of what the EWG is working, stressing this element and providing ways to ensure that trials are conducted in an effective manner, since they're really important for us as evidence generation tools. The EWG shares the perspective that trials should be efficient and robust to inform the decisions of many stakeholders. And the ICH E6R3 is being developed as a robust and responsive guideline that facilitates innovation while protecting trial participants. The EWG is actively working on Annex 1 currently and will continue to focus on risk-based approach to GCP. Finally, again, please consider joining us. And I put the link for our web conference next week in the bottom here, and we hope to provide additional information to our stakeholders at large. With that said, I want to thank the EWG members who actually, all the work I presented today is thanks to them and credit to them. Collectively, an amazing working group that worked together even when we couldn't meet face-to-face and across time zones. So we're extremely thankful for the working group. And this is just a picture of all of us when we met for the first time in Singapore. Thank you so much for listening to me, and I look forward to the Q&A session.
Good afternoon. My name is Ashley Boam, and I'm happy to be with you here today. I serve as the Director of the Office of Policy for Pharmaceutical Quality here at FDA in the Center for Drugs, and I also serve as the Rapporteur for the ICH Q12 Implementation Working Group. In my talk today, I'll spend a few moments to go back just as a refresher on the objectives and scope of the Q12 guideline, to point out some of the key regulatory tools in the guideline, provide an update on status, cover some of the activities of the implementation working group, and then I'll speak a little bit about FDA's implementation progress and activities. So just as a reminder, the title of Q12 is Technical and Regulatory Considerations for Pharmaceutical Product Lifecycle Management. The objectives of the Q12 guideline were to harmonize change management in a more transparent way across ICH regions, to facilitate risk-based regulatory oversight, to better emphasize control strategy and the importance of the development work that feeds into the control strategy as a key component of the regulatory dossier, and to support continual improvement and facilitate innovation in pharmaceutical manufacturing. And then to enhance the use of what might be existing regulatory tools for prospective change management, to enable firms to be more strategic in how they manage post-approval changes. The scope of the Q12 guideline is quite broad. It includes both drug substances and drug products, both small and large molecules that require marketing authorization, which means it includes innovator products, generics, biologics, and biosimilars. It also includes drug-device combination products that meet the definition of a pharmaceutical or biological product. In the U.S. system, this translates into drug-device and biologic device combination products that are led by the Center for Drugs or the Center for Biologics. Q12 does not, however, include in its scope changes needed to comply with updates or changes to pharmacopeal monographs. I'd like to take a few moments to review some of the tools in the ICH Q12 guideline. So established conditions is intended to provide a clear understanding between the applicant or authorization holder and the regulator regarding the elements to assure product quality, and that would involve a regulatory communication if changed. The guideline talks about how established conditions are identified, as well as what information can be designated as supportive information, and so therefore would not require reporting to the regulator if changed. Post-approval change management protocols, which are also known as comparability protocols in the U.S. system, are a regulatory tool that can provide predictability regarding the information required to support a CMC change, as well as the type of regulatory submission based on a prior agreement reached between the applicant or authorization holder and the regulator. This type of mechanism enables planning and implementation of future changes to established conditions in a more efficient and predictable manner. The PLCM, or Product Lifecycle Management Document, is intended to serve as a central repository for established conditions and the associated reporting category for changes made to those ECs. This document will also capture how a product will be managed during the commercial phase of the life cycle, including any relevant post-approval commitments and post-approval change management protocols. Finally, the structured approaches for frequent CMC post-approval changes describe the strategy for a structured approach that is applicable to frequent changes in the CMC space, and a discussion of data expectations, which would then enable the use of immediate or other post-implementation notification for these types of changes, even in the absence of approved established conditions. The ICH Q12 guideline reached Step 4 in November of 2019 in Singapore. You see my very happy colleagues from both Health Canada and the US FDA as they signed off on the Step 4 document. So then moving on to implementation. The different ICH regions are beginning implementation, and the ICH guideline includes text encouraging regulatory members to provide information on their websites about the current status of implementation, specifically with regard to regulatory considerations. Some of the tools in ICH Q12 are new for certain regions, and some are existing. And so this information on the websites will help applicants and authorization holders understand which pieces of Q12 have been implemented in the different regions. The ICH Q12 Expert Working Group was then modified to become an Implementation Working Group, or IWG, and a concept paper for the IWG was approved in March 2020. The IWG has been busy developing global training materials, and we've also engaged with PIC-S, or the Pharmaceutical Inspectorate Cooperation Scheme, which consists of global inspectorates, to help develop training materials for inspectors and investigators as they go out to facilities where established conditions may have been implemented, for example. So as I mentioned, the Q12 IWG has been developing training materials, and while the main focus is materials for ICH regions, we've been putting these materials together with an eye towards them being also useful in non-ICH regions. One part of the training materials will be slide sets or modules that will address each section of the guideline. And these slides, once adopted, will be posted on the ICH website. We are also working on case studies with additional examples and narrative text to provide a better sense for what regulators will be expecting for each of the types of tools, and the examples we are developing are largely based on input provided during the public consultation period as the guideline was being developed. The examples include more situations in which established conditions might be proposed, including for APIs, for vaccine products, for drug-device combination products, and for analytical methods. We also are working on further examples of PACMPs and the PLCM document. And finally, in cooperation with our colleagues in PICS, are developing an example to outline how the pharmaceutical quality system and findings on inspection related to the PQS might impact regulatory flexibility previously granted through use of Q12 tools. In addition to developing the training materials, the IWG has been used as a forum in which both regulators and industry can share experience and learnings from implementation. FDA shared its experience with our established conditions pilot, about which I'll speak more in a few moments, and industry colleagues who participated also shared their experience with the pilot, so both perspectives could be gained. We expect to have additional sharings as the IWG moves forward and as different regions further their implementation efforts. In terms of FDA implementation, I'm very happy to announce that FDA has both adopted and now published the Step 5 guideline this week. It does replace the 2015 draft guidance on established conditions. FDA has also developed a draft guidance on considerations for implementation of Q12 to provide more specifics and clarity on how to implement the guideline within the U.S. regulatory system. This draft guidance is currently awaiting publication. We are also developing internal guidance for our assessment staff. In FDA, we refer to this as a MAP, or Manual of Policies and Procedures, to provide information and guidance to our assessment teams on how to implement Q12. And this document is in progress being developed. FDA has also put in place a number of different steps with respect to training for our assessment teams. And we started back in 2018 to try to both build awareness and get our assessment teams up to speed so they'd be ready for implementation of the guideline. So let me take a few moments first to talk about our established conditions pilot. FDA recognized that established conditions would be a new tool new regulatory approach in terms of having established conditions specified within an application and formally approved. And so we initiated a pilot in 2019 to give both our assessment teams as well as industry an opportunity to try this out, to see how it would work, and then identify where there were additional needs for implementation guidance, for training, and other pieces that might be needed to further our implementation once Q12 was finalized. We accepted 9 applications into the pilot, and you see we had a nice mix of both small and large molecule products, originals and supplements, and we had inquiries about participation from both innovator and generic firms. We've learned quite a lot from our experience with the EC pilot, and those learnings have informed both our implementation guidance I mentioned, as well as the map that's under development. So then back to FDA's training efforts. We started back in 2018, as I mentioned, when Q12 was still very much under development, but we understood that it would take multiple steps to get our assessors up to speed and ready to implement the guideline once finalized. So we started with phase 1 of our training, which was really just to provide awareness to our assessors and to help provide clarity on the intent of Q12 and the core elements or tools that were being developed in the guideline. At that point, we used theoretical examples to help illustrate the concepts and demonstrate for our assessment teams how, for example, established conditions might be identified in an application. In phase 2, recognizing the foundational nature of both the pharmaceutical quality system and cGMP requirements, we provided a refresher to our assessment teams to make sure that they not only understood the requirements within our cGMP regulations and the importance of the PQS, but how those two pieces would be foundational and would support our ongoing assessment of Q12 tools, including established conditions. In phase 3, we gave further training based on the learnings from our established conditions pilot. And in this case, we were able to use real-world examples from the pilot to walk assessors through the implementation process. We were able to use feedback from the teams involved in the pilot to share real-world experiences, what it was like to receive proposed established conditions and associated reporting categories, how to consider those in the overall context of the control strategy, and how to interact with applicants, whether explaining further the information that FDA needed in order to make decisions, how to word deficiencies where additional information is being requested from applicants. Really, how does that engagement work, and how do we get to ultimately a set of established conditions that we could agree to approve? And now that we have published the Step 5 guideline, we will move into the next phase, where we will have a support team available to work with our assessors to help answer questions, to ensure consistent interpretation of the principles in Q12, and to provide oversight to help guide consistency of implementation to make sure it is consistent with those principles. So in summary, ICH Q12 is an important guideline which includes tools and enablers to facilitate innovation and continual improvement, which can help move pharmaceutical manufacturing into a more robust environment that can help reduce supply chain disruptions and ultimate shortages. Implementation of the Q12 guideline is underway at FDA and with other regulators around the world. The ICH Q12 implementation work group will continue to support implementation both through the development of training materials, the first of which should be available in the near future, and by sharing experiences regarding implementation from both industry and regulators. So I thank you for your kind attention, and we'll be happy to answer questions at the appropriate time. Thank you so very much.